before it makes it to the marine website.
Right. No, that was cool. Thank you.
Can I just do a comms check with chart table to make sure we have good headset comms?
That's a great idea.
Can you hear me?
Which chart are you?
Yeah, which chart are we doing? Are you on chart?
I'm on 2.
You're on 2. Okay, let me do that. All right, let's try that. You can hear me, Amy? I can hear you, actually, I think.
Yeah,
Can you hear me?
I think so. Do I need to press down on the Herc?
We can hear you.
So if you click
Okay.
the upper light is lit, then you should be able to hear us.
Okay.
Yeah. So Jess,
Oh yeah, I can hear you now.
You can hear me now? And Jess, you can hear Jess?
Yep.
Yes.
Okay, perfect.
Awesome. Thanks for testing that. Yeah, yesterday it's a little challenging, but
Yeah.
got it sorted out, I think.
Yeah.
Chart 1 should be good as well.
Oh, good. So we've got 2 out of the 3 working back there.
No, I mean all 3 are
Oh,
good.
all 3?
Oh, great. I'm going to slow down a little bit.
Yeah, jumping a bit fast now.
So, Sarah, I had a quick question for you from Nautilus Live. Is now a good time?
Sure.
Yeah. So we can't quite see it at the moment in the Hercules view, but occasionally you can see the craft Predator arm, and you see it's got these 3 colored stripes on it: red, green, and blue. That's just purely for decoration, right?
Oh, John.
That just makes it look pretty, yeah.
Oh, yes. No, actually, it's the colors on the arm are for white balancing, and actually we don't use the color one so much. We use the white one, and so you'll see us when we get to the bottom. Take a second to zoom in on that and set the white balance for the video.
Cool.
And yeah, so that's part of the whole process.
Thank you.
Yeah. Those other ones would be more for color balance, but we usually just do the white balance one.
Okay.
Yeah.
Does that kind of change a little bit each dive depending upon the seawater conditions, whether it's a little bit turbid or cloudy?
It could, I guess. Different. I am not a color scientist of how it might change based on like at extreme depth compared to other extreme depth. But it is different than the surface for sure, because they're, you know, like red lights are filtered out and everything. So, but
they're all
but
on,
I don't know, different depths.
So
it might change a little bit. We just set it because it's good to have that start fresh every time.
Oh yeah. No, that's cool. Thank you.
Yeah. Whoa.
What was that?
Squid just plopped down.
Yeah.
I didn't see it bounce back, though.
we can try it. So yeah, let's do that. Let's turn that off. And I'll turn the bucket on. Right, because they're the same circuit essentially. Yeah, unless it's the camera itself, yeah. That would be the only thing you're checking against by flipping them. Yeah. Yeah. It was like, okay, so you know how you put the bottom loop and the top loop of the lids together in the trigger? And those are only like maybe what, like 1/8 of an inch thick? So it just pulled just enough to pop the top, and the bottom loop stayed in the trigger. So it was like almost impossible to do. It was just a total fluke. It was nothing that went wrong. It was just like a total like fluke on the pull that I've never seen that happen before. So it was just random like luck. Bad luck. That it just was like pulled it maybe like a fraction more and it would have gone. So yeah, that was just unfortunate luck on that one.
Sarah, did you actually see that squid bounce? I was looking down at the moment.
I did. It just
Oh,
like
yeah?
fell right there and very ploppy, but I don't see it.
So for those of you watching on nautiluslive.org, now that we're settled into our descent,
Give her a little more, maybe go to like 26 or something like that. Sorry, John.
no, it's okay.
Roger.
We can take a little time just to orientate you on today's operations. Nicole Reynaud, our watch leader, gave you... Oh, another
Man
squid.
alive.
There
I
we
wanna be
go.
caught.
I actually saw that one.
Still there.
Oh, yeah.
Oh, there's one right there on the porch. Look down.
Yeah, go look for it. There it
Oh,
is.
yeah. Nice.
Remember it when it attacked the craft? Were you over there when it attacked the craft?
No,
It was on the craft, like turning all crazy colors and like super mad. And it was just all over the craft arm, just like trying to eat it.
I don't think we have any more friends here.
Yeah. That's actually a question we get a lot about animals interacting, and I'd only ever seen the, what was it, the octopus that wouldn't let go of one of our tools before.
Right, yeah.
But turning crazy colors is pretty good. Maybe they're all sheltering from the sperm whale that's hiding behind Hercules at the moment.
Don't give us false hopes.
No, I know. I know.
They're already.
It's a pretty one too.
Yeah.
There's some wonderful sea creatures on this descent.
Ocean's alive.
This has been one of the more active water columns
Yeah.
that I've seen in a long time.
So I just wanted to make a quick comment for those watching on Nautilus Live, just to let you know that our ROVs, Hercules and Argus, they're currently descending through the water column. We are 300 kilometers off the west coast of North America, diving above the Gorda Ridge tectonic feature. This is where the Gorda and Pacific
on
Plate.
Argus is getting spotty.
This is where the Gorda and Pacific Plates meet each other. They're actually spreading apart at this point on the Earth's crust, creating a weak point. And that weak point manifests itself as a gigantic submarine rift valley. Along this kind of central axis of that crack, the actual fault itself, there's a lot of earthquake, volcanic and hydrothermal activity. We're going to be just operating on the steep sides of that rift valley today at the Seacliff hydrothermal vent site. Yesterday we had a really busy day encountering that site for the first time in 13 years, performing some preliminary mapping and temperature measurements. And as Nicole was mentioning earlier, we've got a full day of experiments at that site today.
You can rack out and get rid of all the...
Get rid of all that stuff.
Yeah.
Looks like we're collecting a few cobwebs on the way down.
They forgot a dust.
I just want to say a quick hello to Justin Richards and his physical sciences class in Roebuck, South Carolina. Justin is a science communication fellow who will be joining the Nautilus, I believe, on a cruise later this summer. He and his class are watching and getting fired up. So another great question on Nautilus Live we just had is, do these hydrothermal vent features that we are diving down to investigate, do they affect the water column above them?
Yes.
Yeah, I was going to
They
ask
do.
if somebody wanted to jump in and answer that one.
Well, there's been a neat publication with this data, the mapper data, which we have 2 mappers, or instrument suites. Miniature autonomous plume recorders from NOAA PMEL, Pacific Marine Environmental Lab. And these have a series of sensors including temperature, EH, potential turbidity. Anything else? Do they have salinity? Yeah, depth and pressure source standard, stuff like that. But scientists who've visited this area with those sensors have basically been able to tell the volume of the plume from driving sort of the sensors around. We have one on Hercules' light bar right now, so that's where we're going to get the measurements from on this dive. And so you can see where basically the background water versus the plume water, you can see the changes. And yeah, then just kind of by doing a survey through it, you can see 3-dimensionally how extensive the plume is. And at this site, it's fairly extensive.
Okay.
It rises both up in the water column, but also extends laterally over the sites.
And these plumes, they typically rise up several hundred meters above the vent site, is that right, till they become neutrally buoyant?
That's right. It's usually one way early scientists still can detect where hydrothermal vents are. You know, there's small point sources, so it's hard to, it's sort of almost chance if you see them, right?
Yes.
But if you can use another sensor to locate the plumes, then you can sort of hone in on them and find where those temperature anomalies or differences in sort of the fluid are coming from.
Yeah, I remember that's how the first hydrothermal vent systems were discovered. It was towed sledge surveys with temperature sensors on board.
Yeah, that's true. And yesterday, actually, Megan was keeping a close eye on the CTD on Hercules to note when there were even small anomalies, like a 1/2 degree, 1/4 degree. So we knew that we were getting close to the vent sites when she noticed those spikes in temperature.
Oh, that's cool. Thank you.
Yeah, for people who are just tuning in, this is the second dive at North Gorda Ridge for us at what's called the Seacliff vent site. Yesterday we had a 16-hour dive that was pretty successful. We were able to locate the sites that hadn't been visited in over a decade. We did a visual survey to mark the perimeter of the site, which was looking for the area where the chimneys were, and then there are ciliate mats and diffuse flow, and then tube worms on the perimeters of these sites. So we were looking for all of those visual cues and spent some time doing that. We dropped off some microbial experiments that are at a site of diffuse flow and spent some time with a high-resolution mapping system on Hercules getting a detailed map of the site so that the scientists who are on shore at the Inner Space Center can look at the features and make decisions about future dives. And then with the final amount of time we had on that dive, we did some temperature probes to determine whether the temperature at this site has been stable over the last decade since scientists have been here.
So one thing I really enjoy about these what we call blue water dive or blue water shifts when we're basically spending a few hours getting to the seafloor. We can actually spend a bit of time on Nautilus Live, going backwards and forwards with you guys, posting questions on nautiluslive.org. There's always a special place in my heart for elementary schools watching us. So we have Lawton Childs Elementary School from Florida who are watching us right now. And I'm just going to throw out their question to the room and see if anybody wants to dive in. What is the strangest or most unexpected thing the team has seen underwater?
For me, I would say the gulper eel last year, because I was in the mess where we eat meals and saw it on the screen and just was stopped in my tracks. I didn't know what it was. And when you could find the highlight video for gulper eel online for Nautilus, and you'll see that it makes an amazing transformation. So it didn't look anything like an eel, and then all of a sudden it transformed into one.
Yes.
It's very unusual. And then one of the really incredible things about that too was one of the students who was out here identified it. So she was our data logger, a returning intern, and she knew what it was.
Wow.
That was pretty cool.
How about the front row? You guys must have seen it all. Seen one gulper eel, seen 1,000.
I never saw the gulper eel, so I was kind of disappointed I missed that one.
Yeah.
I don't know. It's hard to pick one thing. I see lots of really cool things down there, so it's difficult to pick one thing. At the end of last season, that octopus garden area, that on the, I think it was the very last cruise of the season, was really amazing. That's another highlight one on the Nautilus Live website that you can find. It was really impressive. And then I remember, I can't remember the cruise exactly, but I remember just in my mind we were doing some cliff work and doing a lot of video of the biology that was on there, and there was some biology they were looking for, sort of a little sparse here and there were patches and stuff, and then we just came across this sort of arching rock form that made an arch, and the amount of the density of life living on that just strip was just unbelievable. It was obviously the currents were taking food in, swirling it in and around that rock formation, and it just made like 110% coverage of animals living on this sort of archway. And so we took a lot of time and did a slow transect filming all this whole thing up and around, and that was pretty incredible. That was in this area. I think it was just north of here.
Okay.
That we were doing that. It was pretty neat. I have to recall the actual cruise, but it's slipping my mind right now.
Yeah. I was pretty stoked yesterday that we found a miniature octopus garden at our hydrothermal vent site.
Yeah. There's quite a few there.
Yeah.
Those were the first octopuses I saw on watch before. Last year I didn't see any.
Oh wow.
Okay. Were you on that cruise for the Davidson Seamount?
No.
Okay, right.
But everybody's always excited to hopefully see an octopus.
Oh yeah.
We're always like, oh, we hope we see some octopuses. Is that right? Octopuses? Octopi?
Octopuses.
All right.
Yeah.
Phew. Octopuses. All right. But we never did. Not on my watch, at least. Not on my watch. And so yesterday was exciting for me to see those.
Oh yeah.
For the first time down there in live, you know.
Yeah. So you actually have, Sarah, you have a background as a wildlife filmmaker, is that right?
Yeah, that's what I am in my non-Nautilus time. I film other wildlife.
Mainly on land?
Yes.
Okay.
So yeah, but you know, there's nothing like the deep blue.
The deep blue. That's profound. That really is profound.
Really? Oh.
There is nothing like the deep blue. Yeah.
No, it's just cool.
That's cool. Yeah.
Because you never know what you're gonna see.
Yeah.
And it's, you know, either hasn't been explored in years, or we're exploring it for the first time, or we're exploring it with new technology.
Yeah.
Or something like that, and it's always cool to
Yeah, I'm just
see what's
interested.
out there.
I'm just interested in kind of the inspiration you must get as a filmmaker, because we encounter so many interesting stories down here. And do you ever kind of see something and store it in your brain and think, I'd like to come back to that someday?
I'd like to come back to all this, but it's difficult when it's 3,000 meters below the surface.
Yeah.
But yeah, it is cool looking through it in a kind of storytelling lens,
Mm-hmm.
and it's just really interesting seeing how it all fits in with, you know, the rest of the world when I like have a background in wildlife and stuff, and it's cool to see how life is just so different in different parts of
Oh, yeah.
Earth.
Yeah.
It's neat. This is on our planet.
So just for those of you on Nautilus Live wondering how long it takes us to get down to the sea floor at these depths, we'll be operating at a depth of somewhere between 2,600 and 2,700 meters today. Yesterday, I think it took about 2 hours and 40 minutes or so, 2.5 hours to get down to the sea floor. So that's just a good number to have in your mind. We'll have a better idea, obviously, as we get closer, but given that we launched pretty much at 8 o'clock this morning, it might be somewhere around 10:30, 11 o'clock we might be touching down.
Yeah, we are currently 70 minutes from the seafloor, 70.
70, hour and 10 minutes. So that's pretty quick. That's going to be about 10 past 10. That's not bad. Are we carrying any of those big, heavy microbiological cultivation experiments today?
Thankfully, no.
Right.
Those are all still down
Right.
on the seafloor. Yes.
Hopefully
yeah,
we're
hoping
reluctant.
our ballast is a little better today.
Yeah. I was just wondering, could we take a quick look at the porch of Herc and see with the camera, one of the cameras, and just see what we've got on board today?
Sure thing.
Okay.
We got some wands, we got a crowbar, we got a temp probe.
Is that the crowbar of science?
Crowbar of science.
There we go.
Crowbar / pokey stick.
Yes.
/ pokey stick.
Oh, I see. You've got the length measurements on there as well for sticking it into the sediment.
Yes.
Right. What's in bucket A?
Those are markers.
Those are markers.
Okay.
Yeah. And then we have some other little treasures in the in our front cupboard, but we'll show those later. Those are secret. You have to stay tuned in to find out what's inside the drawer.
And then one and two, those almost like coupled metal cylinders. Those are our IGTs, or our isobaric gas tights.
Those are them.
I'm looking forward to seeing those being used today. I might be in the lounge when they're being used, but these guys are basically, they're going to sample vent fluid coming straight out of these vents, but the neat thing is they keep it under whatever it is, I guess 270 atmospheres of pressure all the way up to the surface, and then under controlled conditions in the lab we can actually sample that fluid and all of the gases that remain in solution. And it's actually a really neat probe of the water-rock interactions that are happening deep below the surface of the hydrothermal vent site, where we can't actually penetrate to with any kind of direct measurement, but by looking at the chemistry of the vent fluid, we get a really, sometimes, an improved understanding of what kind of reactions are happening as the water descends through the faults and is then heated close to the magma chamber underneath. So thank you for all of your questions. You guys are continuing to post on nautiluslive.org. It's actually great looking through the feed as it comes in. The other great thing is, especially on these blue water watches, I've got a room full of experts that I can just bug all the way down. And another question we've just had coming in is, what is the hottest vent you have recorded, or that anyone remembers, and where was it?
That's a tough question.
I've been close to, I've been in the 300s before. I melted a core tube once. I don't know what that is. Yeah,
Where was that, John?
that was in
That's
Fiji.
cool. Wow.
Yeah, there's some really hot vents over there, and we sort of stepped off to the side to try and do a core tube near some venting area, and stuck the core tube in and pulled it out, and it was all melted.
Oh, that'd be cool.
So we decided to get off the bottom at that point.
Yeah.
Do you know, Josh, do you remember how warm Endeavour is?
Yeah, it can range, but it can get into kind of like into the 300s,
Wow.
I think, high 200s,
Yeah.
a few spots, yeah.
There's a great video on the Nautilus YouTube channel of exactly that, and there's a lovely segment where they've got the temperature probe in a vent, and everybody in the room is narrating the temperature that we're getting, and that was about 327 degrees Celsius.
Wow.
Yeah.
Which one was that? Was that Guaymas?
No, that's Endeavour.
That is Endeavour. Huh?
Yeah. For those of you who don't know where Endeavour is, that's actually just a couple of 100 kilometers north of where we are. I mentioned that today we're at a location on the planet where the Pacific Plate meets the Gorda Plate. Just north of the Gorda Plate, off the coast of Washington State and Vancouver Island, is the Juan de Fuca Plate. And it's thought that all of these kind of little broken plates are essentially the remnants of a larger plate that fractured. But yeah, and Endeavour is just at the junction of the Juan de Fuca and Pacific Plates. Another one of these, that's a black smoker hydrothermal vent site.
Yeah, the site at Seacliff that we're going to has had a temperature at the surface reported of 308 degrees Celsius.
Right.
And they expect that the temperature at depth in the seafloor is well over 400 C.
Right.
Wow.
And so we'll be, part of what we're doing is looking at the long-term stability of the temperature of the fluids. It was unchanged between 2002 and 2004 when researchers were there.
Uh-huh.
They didn't measure the fluids in temperature in 1985 was when it was discovered, but they expect that it's been pretty stable over the course of the last couple of decades or several decades now.
Yeah, I sat in on the afternoon watch yesterday, and I think the highest they got was just over 200 Celsius.
Oh wow! Yeah, I didn't know that. But we'll do a bunch more measurements, I think, in the coming days.
Yeah. It really shows, those temperature measurements really show some of the capabilities of Hercules, because a movement in the wand of about a centimeter around the aperture of the vent, the opening of the vent, you completely changes the temperature measurement. It's amazing how precise you have to be. So for those of you on Nautilus Live just wondering how long it's going to be before we encounter the seafloor, looks like at the moment it is 9:07 Pacific time. We're estimating perhaps 1 hour from now, thereabouts. Which is actually a pretty good rate of descent. In my mind, I always had kind of an hour per 1,000 meters.
Is it possible to slow a little bit?
You can speed up if you want, yeah. Just catch up a little bit. I haven't stayed pretty steady.
All right.
Right now we're doing about 27 meters per minute.
Jamming.
Do you know how to put the temp probe thing up on the sonar screen? Do you know how to do that?
Yeah.
That's pretty good. That's about 1.6 kilometers every hour.
One of the interesting things I read about this hydrothermal vent field is that it's one of 2 in the US EEZ.
Okay.
Can anyone name the second?
For the
Trivia.
hydrothermal vent area?
Yeah.
In
the US EEZ. Hydrothermal.
the whole US?
Warmer than, what are we saying, 120? Something like that. In the whole US. So last year we were at Loihi, right? And that was not considered hydrothermal.
Okay. So EEZ, that stands for Exclusive Economic Zone, is that right?
That's right.
Okay.
And I'm assuming axial is not, right? Wouldn't
Axial's
be considered
not.
hydrothermal.
Actually, I'll give you a clue. It's very close by.
Is it?
And many of you probably read papers on these associated vent fields. So it's Escanaba to the south. And this was
Escanaba.
according to one of the papers I
okay.
read. So I
don't think I've quite been there. I've been to axial a few times. So that's an
Yeah.
I'd love to have an axial.
I think
So
Escanaba is
cool.
less studied than even this Sea Cliff site. So it's kind of interesting. It's a much more sediment-rich site. It's more similar to the Guaymas basin vents that we went to a few years ago in Mexico and Baja.
Oh, that's surprising. There's only 2.
I am too. Yeah.
Yeah.
Huh.
And
it's
perhaps
120 degrees is the.
that's what I think we're defining as like
defining
the temperature.
But the EEZ, is that 200 nautical miles?
It is, yes.
Right.
I can't think of another. I was trying to think if the paper might be outdated because it was published maybe a decade ago now, but I don't think there are others.
Right.
Yeah, we're starting to lose our Argus. Yeah. Starting to suspect the cable between the camera and our junction box, because we have known good camera. The other camera, which didn't work great, is now on 1. It's solid, and we're starting to. We saw flicker at like 700, and now we're seeing it's getting worse.
Yeah.
1270. So probably, and then we know all our kind of wiring and circuitry is good everywhere else. So
Yeah.
probably the cable between the camera and the junction box. Because the dash cam's been solid the whole time. That was the Argus spot on the previous dive. It's getting squeezed.
So one of the neat things on nautiluslive.org during these long descents through the water column is we get a chance to talk around a number of themes. I've got a great one I'd just like to pass over to you quickly, Sarah, next to me, if it's a good moment. We've actually got a viewer, Finn, from the UK, who's 15 years old and is trying to get into nature photography and perhaps wildlife photography. And I know that part of your education has taken you to the UK and to a pretty good school in the UK, and I wondered if you wanted to talk a little bit about that experience.
Sure. Well, Finn, you're lucky because you're in a good place for it. For the wildlife filmmaking kind of industry there. There's a lot of it in Bristol. And I went to the University of the West of England, and I got a master's in wildlife filmmaking there. So shout out. But it's a good, I mean, it's a good program. And yeah, Bristol's a city in the world that has probably more wildlife filmmaking than anywhere else because of the BBC Natural History Unit and a lot of other production companies that are there now as well. And there's just a lot of great documentary natural history filmmaking that comes out of there. So Finn's lucky.
Yeah, no, that's good advice. Thank you.
Yeah. I'd say, I mean, Finn could just, anybody, just, you know, practice and keep doing it. I got my undergraduate degree in wildlife ecology, so I also like having the science background.
Oh yeah, yeah, for sure.
Knowing animal behavior and just how things work.
And how much experience did you kind of need to be accepted for the? Because you did a master's in wildlife photography or wildlife filmmaking.
Filmmaking, yeah.
Yeah. So what background did they look for in terms of the people they selected?
There's a mix. It's not a huge number of people in the course any given year, but it's a mix of people with film backgrounds or people with scientific backgrounds.
Mm-hmm.
And so part of it is learning from your peers as well, just because everybody has different experiences and knowledge. But everybody, like, the commonality has been that everybody's been involved in both wildlife and film. So, like,
Right.
if you've had a majority wildlife background like I did, I still tried to, like, involve myself in filmmaking and do it when I could and practice it. And so that's what they look for.
Right.
Not just being able to tell them you have an interest, but being able to show them with your, I don't know, maybe extracurriculars or just things you've made yourself that you have an invested interest.
No, that's cool. Thank you.
So, do you do any video work with film, or is it all digital in filmmaking school?
It was all digital. And the program I went to was a 1-year program, so it wasn't like a whole like history of film. It was very specific to like the storytelling side of things, knowing the industry and all the different, what is that?
Big red.
That's some wildlife.
Wildlife! Science! Yeah, so it's really specific to like things that are happening right now. It's very current.
Film can happen now.
No,
right?
it can happen now, but it wasn't like a
Yeah.
like a I don't know, if you go to like a 4-year university to study film, it would probably be a more like, you'd have classes on like the history of film and you know, back from when it started and have all the kind of detail of that. But this is very specific to wildlife filmmaking and kind of giving people the knowledge of the industry as it is, and of course learning about what has been made and how it was made, because it's not like we don't use that at all. But yeah. So I use digital.
Sweet. Sounds good.
For wildlife filmmaking, digital really has helped a lot, because imagine carrying rolls of film through, you know, storms, or, you know, carrying it anywhere at long distances. You don't have ginormous crews, and you know, if people are in the rainforest or the ocean, you know, trying to film whales, I mean, it's just so much, there's so much harder to manage actual film.
And yeah, I mean, we couldn't have telepresence without digital, so
Right.
our whole operation here wouldn't really exist in the way it is now.
Yeah, and there are a lot of live shows now, like Nautilus Live over in the UK. They have Spring Watch and the watches, and just you couldn't do that either. So
Well, I'm just going to duck out of the room for a little bit. I'm off to talk to Journey Middle School in Souk, BC. Have a quick chat with the students there about science. Susan Groff, another of the science communication fellows on board, she's going to take my seat for a little bit. So I'll see you guys in about 40 minutes or so.
Great, thanks, John.
Toodles.
See how you're pulling Argus away from the ship? So you could just back off or even shut off the forward stick lock. Hey Nicole, can you remind me what the first move is once we get down at the bottom and settled, where we're headed to?
Sure. I mean, aside from the first things being just our usual tests and,
Yep.
and checking the super,
Yep.
we're going to head uphill to waypoint 1, where the colonization chambers are. So yeah, just, and yeah,
Oh,
Alex,
I see.
if you want to measure that
NA108-H1751.
distance, that would be useful information. Because one of the things I was going to ask as we continue our descent is if we wanted to move the ship any further west to position us to just start our ROV moves right away. Or if you guys like being pulled off this distance, knowing that there's a bit of a hill.
Right. Yeah. Well, I think we just turned the Herc was kind of pulling a little bit, so we probably will start swinging back.
Okay.
Waypoint 1. Oh, that's waypoint 1 up there.
Oh, you guys are in
330.
feet. Do you mind changing the meters? Thank you.
100 meters.
Okay. Whatever you're comfortable with, Josh and Jess.
Yeah. I'm gonna see what happens now that we're not pulling anymore. We might get pulled back, and then yeah, we could step a little bit more to the west to that launch position. So Argus is a little more over that launch position, but I think it'll start making its way back there right now as well. We'll keep an eye on it, but yeah, I agree. We should try and land there and then make our way.
And when you say there, Josh, I just...
Oh, sorry,
land
at the launch,
at the launch. Got it.
targets.
Okay.
So acquire bottom at the launch target.
Yeah,
roughly.
and then we'll just make our way up.
Sorry, be more clear in my words there.
It's okay.
Hi, Susan.
Maybe you should slow down a little bit on your descent. You're at like 32 now.
Ooh, yeah.
Okay. Could someone answer this question regarding the density as you get further out versus close to the shore? Does the density of the water change?
Well, there's certainly layers with different density in the ocean. And as seafloor mappers, Alex and I and Megan and those of us who've done mapping before know that that impacts our ability to map the seafloor. So what changes most that affects density in the seawater is the salinity and the temperature and then pressure at depth. So those are the three factors that will affect the water density. And you have a very steep change in those properties close to the surface and in shallow areas. And then you have a very homogeneous, or most places in the ocean have a very homogeneous layer, maybe, 50 meters or more below, where things change either very slowly and then below a certain depth don't really change at all, measurably. So the way we figure that out is by taking a CTD with us, which measures salinity and temperature with depth. And so we could see what those properties are and then figure out what the density of the seawater is.
Thank you.
So I think what we'll do when we get closer to the bottom is let's all stop like 50 meters above the bottom. And since you're already kind of pointed in the direction that we're going to go, you can back up towards me and then I'll turn around to find you, and then we're set, and then we can come down and find the seafloor at that point. Yeah, Argus has already swung back to the launch position.
It has, yeah.
Alex Wang, could you possibly share a little bit about your role in with Nautilus? And you're from the Naval Academy, correct?
That is correct. On the Nautilus, I am the navigator. That role entails being essentially the liaison between the bridge, are the people, the crew that are actually driving the ship, and the science team and the ROV pilots. So Argus is tethered to the ship, and my role is to move Argus around so that Hercs could go down and do whatever it needs to do at the seafloor. I'm also the eyes and the overlooking, making sure, you know, everything is basically the eyes on the surface, to be in charge of the spatial awareness. Yeah.
Thank you. Appreciate it.
Susan, can you tell us about yourself?
I'm a high school biology teacher in Southern California, and I have an interest in astrobiology, and I thought that applying for this Nautilus Science Communication Fellowship would be a wonderful opportunity to explore this further. So I'm happy to do that. Thank you.
Yeah, welcome.
Thank you. So Amy, could you possibly share with us about what temperatures you're looking for today in bacteria or archaea?
Sure. So we're looking for low temperature venting, and that generally for us means somewhere between like 15 degrees Celsius and 50 degrees Celsius. At least at the first vent site, that's what we're hoping to get. And partly that's because eukaryotes tend to not like high temperatures, like 80 or 100 degrees. So we're trying to capture that community as well at this first site. And Sarah Hu, who's also here with me from Hui, is interested in that community. And so she's going to study the eukaryotes and how their grazing might impact the microbial community. And I'm looking at the bacteria and archaea in the fluids. And the second site is not as constrained. We could go a little bit higher or lower if we needed to for the temperature. But yeah, I mean, it's still pretty hot. You know, 50 degrees Celsius is still pretty warm, but anything over 120 degrees you do not expect to have life in, because that's kind of the limit that we know of. So we're looking for the lower temperature vents today because we're sampling for microbiology, and I think in some other subsequent dives we'll be looking at the high temperature fluids as well to try to get a handle on the chemistry of the system and the energetics and what types of energy sources are available for microbial life in this type of environment.
Thank you. Sarah, could you possibly share with us how challenging it is to maintain the cultures for when you take them back to the lab? What are some conditions?
Oh, she's not in the lab van right now.
Oh, okay. Perhaps, do you mind?
She's, can you ask the question again? Sorry.
How challenging is it to maintain the cultures that are taken from the hydrothermal vents and then brought up to the surface? Are there certain like pressures that they have to be in, temperatures?
Yes. We oftentimes don't worry too much about pressure, but there are a lot of groups of scientists that are looking at the effect of pressure on microbes in the lab. We tend to keep our culture tubes anaerobic and overpressured as well, so try to minimize any impact between the anoxic subsurface, you know, deep pressure, and then the surface. And it is very difficult, and a lot of times you can get a whole, like if you inject some vent fluid into a culture media and you're trying to enrich for a certain type of organism, oftentimes they will grow that first time, but it's already a mix of organisms and they might need each other or might need some metabolite that some other organism makes. And then once you start to transfer them and try to isolate them, it gets more and more difficult down the line. So a very, very small percentage of what we are able to culture in the lab is representative of what we have in these environments. But there are definitely lots of people who are looking at new ways to try to get at those microbes and how best to try to culture them. And that's really the best way to learn about these organisms. You can learn about what they're capable of, so what their genetics can tell you about them, but we really don't know if they're using all of those genes or pathways that they have, and in what circumstances they might be using them. So we couple it with some experiments in the lab to see who might be fixing carbon. And yeah, so we do lots of experiments and culturing, and we try to get at the metabolisms of these organisms from several different angles. And we're usually able to produce a complete story. And it's always really interesting, and every place is different, has different organisms and different metabolisms and energy needs.
Thank you. Is Dr. Lim available today?
She's not in here.
Okay. Is there anyone who's worked with extremophiles in Antarctica? And then
I mean, I've worked with some cold-loving microbes, but not from Antarctica. I've isolated bacteria from lava tube ice caves that have ice all year round in Oregon, and trying to get at the community that lives kind of at the rock-ice interface where there might be a little film of liquid water. And that was mostly for researching organisms that might be found on Mars or might have been able to live on Mars at some point. And those organisms really like it cold, like 4 degrees Celsius out in your fridge, basically the temperature of your fridge. And they actually were kind of easy to grow, I think, but they were also very common lineages of bacteria, like Pseudomonas. And yeah, so I think it's probably more difficult to grow the higher temperature bugs than it is the lower temperature.
Okay, thank you.
You're welcome.
What are they?
PU. Just PU.
Jessica? Hi, Jessica.
Hi.
Could you please share, I've been watching you work on the Hercules ROV, so could you possibly share your background, also how you became involved in engineering?
Sure, yeah. So I became involved with the Nautilus as an intern back in 2015. At that time, I was finishing my bachelor's in biological engineering at MIT. And since then, I guess my other background is I have a master's in mechanical engineering and underway for my PhD. How I made my way out though is through the internship program, and if you love it, then it helps.
Well, I can tell you love it.
I do. This is true.
That's clearly obvious. Yeah.
So, yeah, that's how I got out here and a little bit about my background. But I think it also helps that if you like the science too. The science is really fascinating, and yeah, so if you like the engineering, you like the science, then it's kind of a nice marriage of the two.
And you have a great support group around you. I've watched that, the whole group.
Yeah, everyone rocks.
Absolutely. Mm-hmm.
Yeah, it's a good team.
Josh has been putting up with me since 2015 too.
Yeah.
Look at that.
Woo-hoo. Slowly taking over.
Matthew, could you please share what you would be able to share about the studies and in general at maybe Johnson Space Center, some of the different NASA institutes in astrobiology?
Hi, Susan. This is not Matthew. Matthew was on the earlier shift,
Okay.
which didn't happen, but he'll be back in the van later today. And Matthew Miller is at Johnson Space Center, and he can talk some. I mean, he can answer your question when he's back.
Okay.
Unless he's in the lounge right now and he hears this, and he can jump on the SPL and answer your questions.
All right. Thank you. I apologize.
No, no problem at all. We've got that wall between us, so it's not always visible who's back here in this seat.
Well, who is available that is from Subsea that could talk about maybe what is available at all the different institutes? Maybe Zara?
Well, yes, hi. So I can talk a little bit about some of the NASA research centers. Subsea, of course, is a NASA and NOAA project. The main source of funding comes from a NASA P-STAR, which is a proposal that was headed by the PI, Darlene Lim. And just to give I think what the discussion that you're bringing up that we've been talking about is how the various NASA research centers have different areas of specialty.
Mm-hmm.
So on this NASA team, we have team members who are from NASA Ames Research Center in Mountain View or Moffett Field, California. And then we have Matthew Miller, who is at Johnson Space Center. We also have a team, the technology team, who's currently at the Space Center in Rhode Island. They themselves are also from NASA Ames Research Center. And to just give for a difference between those two centers, Johnson Space Center is where the astronauts train, are selected, mission control is located. Space flight is examined from, certain areas of space flight are examined at Johnson Space Center and other areas are examined at NASA Ames. So depending on what people are interested in, there would be a number of NASA research centers to learn more about those things.
Thank you. I know they have wonderful resources for teachers and the public in general. Mm-hmm.
Yes, the Subsea, if you go to a search engine and